JCraigVenter, Life at the Speed of Light, From the Double Helix to the Dawn of Digital Life. Step into the fascinating world of genetics and embark on a journey to uncover the intricate relationships between DNA, life, and synthetic organisms. The book Life at the Speed of Light, From the Double Helix to the Dawn of Digital Life, is a remarkable exploration of the developments in genetics research, starting from Erwin Schrödinger's revolutionary ideas to the groundbreaking work of scientists like James Watson and Francis Crick. Get ready to dive into a narrative filled with surprising discoveries about DNA's structure, functions, and the potential of artificial life. Gain insights into the possibilities of synthesizing DNA, creating an entirely new kind of life, and even biological teleportation.
Revolutionizing Biology: How Schrödinger's Question Opened New Doors. Erwin Schrödinger's book, What is Life?, posed a question that led to a revolutionary change in the scientific world. Schrödinger, a Nobel laureate, suggested that biological life can be explained solely through physical and chemical processes. His ideas inspired James Watson and Francis Crick's work and led to the discovery of the genetic code for human life. Since then, scientists have worked to unravel DNA's mysteries and comprehend the entire genetic code, an assumption that Schrödinger suggested decades ago. The debate on artificial life. The concept of creating artificial life has been debated in the scientific community since the 19th century. German chemist Friedrich Wöhler's discovery that organic and inorganic materials are not fundamentally different led to discussions on whether life is determined by cellular processes or if it requires a soul or vital function. While the question of whether we can produce life artificially is no longer as pressing as it once was, society still questions whether we should. Fears of creating something beyond our control or facing punishment from a higher power are common. Despite these concerns, exploratory science continues in genetics, genomics, and computing. Decoding gene splicing. Through the discovery of restriction enzymes, gene splicing has allowed scientists to cut and paste genes into foreign DNA, leading to a better understanding of genetics and uncovering the role of RNA in gene replication. Gene splicing has been a significant breakthrough in genetics since scientists discovered how to use certain proteins to cut and paste DNA from one organism into another. These proteins, known as restriction enzymes, serve as chemical scissors to cut a piece of DNA out of a strand, creating a gap that can then be filled with a new piece of DNA. The process of gene splicing has led to significant advancements in the science of genetics, allowing scientists to study the genetic code of organisms in greater detail. Gene splicing experimentation rapidly advanced, using more complex organisms, such as mammals like mice, to uncover new discoveries, including the role of RNA in gene replication. RNA acts as the delivery boy, transporting code from DNA to cellular protein factories that produce proteins. Through gene splicing, scientists also discovered how genetic defects can result in hereditary conditions such as cystic fibrosis. Gene splicing has revolutionized the field of genetics, leading to a better understanding of DNA's role in biological life and uncovering new discoveries about genetic replication. The quest for synthetic life. Geneticists' ambitions of sequencing DNA were brought closer to reality with the introduction of automated DNA sequencing machines powered by advanced lasers and fluorescent dyes. These breakthroughs allowed genetic code to be stored digitally, bringing scientists closer to discovering the constituent parts crucial to all life. The authors' team became the first to completely sequence the DNA of a living organism, laying the groundwork for the quest to produce a synthetic cell. By comparing two different species groups, the team identified 480 common genes essential to all living organisms. To test the theory, an entire chromosome composed solely of these genes was chemically synthesized. With advances in splicing and storage, the stage was set for synthesizing DNA itself. Synthesizing life's building blocks. A team of geneticists set out to produce a complete chromosome based on computer code, taking the virus Phi X174 as an experimental model. With the automated sequencing and synthesis of the DNA code, they produced viable genetic information confirming that biologically active life could be created purely from chemical DNA. The Phi-X174 virus, known for its simple structure, was chosen for the experiment, with the synthetic DNA assembled using specially designed, carefully selected building blocks in the correct order. The research team were then able to confirm that their artificial virus had successfully infected the bacteria host, proving that synthetic DNA contained the information necessary to produce a virus. Overall, the experiment held groundbreaking implications, as it offered a new and detailed understanding of the genetic makeup of life and how it could be replicated and adapted in future scientific ventures. Creating life in a lab. A team successfully synthesizes a bacterial genome, paving the way for the creation of a living, synthetic organism. In a groundbreaking achievement, a team of scientists successfully synthesized a bacterial genome, marking the first time the genome of a living organism had been artificially created. The team's next challenge was to transplant a synthetic genome into a cell to create a living, synthetic organism. To accomplish this feat, the team selected the Mycoplasma genitalium, the tiniest known genome that is part of a living, self-replicating cell and causes urinary tract infections in humans. They accurately synthesized the 582,970 base pairs of DNA by dividing the genome into 101 segments called cassettes, which were independently synthesized and reassembled. To ensure proprietary rights to their work, the team inserted watermarks in the code, specific gene sequences marking the genome's lab of origin. Connecting the different sections was made easier by each cassette containing an overlapping sequence at its start and finish. The final challenge was to find a stable environment for the code, which they found in yeast cells. The scientists then injected the cells with the new DNA, painstakingly examining the DNA sequence and identifying the team's watermark to confirm the successful, synthetic production of a bacterial genome. This groundbreaking achievement opens up the possibility of creating a living, synthetic organism, which will be the team's ultimate project. The ultimate goal of genome synthesis. The scientific community was skeptical about the viability of a synthetic DNA-created organism until it was transplanted into a living cell. The team working on the project abandoned the slow-replicating M. genitalium for a rapidly reproducing M. mycoids. Polyethylene glycol helped in making the host bacterium's membrane more permeable and protected DNA as it was transplanted into a suitable host. The success of this experiment was a significant step towards transforming one species into another. From computer code to living cells. A team of researchers created the first living, self-replicating species to have a computer for a parent. They digitized biology and turned DNA's chemical code into a computer's digital code. Using the polyethylene glycol method, they succeeded in their initial transplantation tests. However, during the final sequencing, synthesis, and transplantation of the M. mycoids genome, a one-letter deletion in the base pair DNA sequencing threw off everything that followed. After the team corrected the sequence, they conducted a subsequent transplant without issues. The newly inserted DNA created cell colonies controlled by the synthetic genome. They even encoded the lab's email address in the synthetic DNA sequence. The team called the new cell M. mycoids JCraigVenter 1.0, in honor of the JCraigVenter Institute. With this experiment, synthetic life became a reality, but people's reactions and the long-term impact of artificial life remain to be seen. Synthetic life, a breakthrough rife with controversy. The creation of the first synthetic life aimed to disprove vitalism and establish the supremacy of DNA. However, critics were divided, some calling it profound, while others dismissed it as haphazard. The use of a natural cell for transplant was also a point of contention for some. While the creation of organisms made entirely from scratch seems an impossible feat, technology's advancement raises concerns over bioterrorism. Hence, regulation is necessary. Nonetheless, the benefits of increased access to technology for biology students and the potential possibilities that digitizing and synthesizing DNA unlocks are undeniable. Teleporting biological information. Earth-based scientists are exploring how to convert genetic information into electromagnetic waves, which will allow biological teleportation of DNA. This technology can be used to transmit information required for building life, which opens up new possibilities for health applications and understanding life on other planets. With robotically controlled genome sequencers, we can read the DNA of any microbe and reconstruct it in a laboratory on Earth, and this could be especially useful for analyzing alien life forms.
The quest to understand life at its most essential level has led to amazing scientific breakthroughs, forever changing the way we understand the world around us. Life at the Speed of Light not only recounts the history of genetic research, but also brings you to the exciting frontier of producing synthetic organisms created from computer code. While the ethical and moral implications of this research pose questions for many, the potential advances in medical and technological applications are truly extraordinary. From creating tailored medications to exploring life forms on distant planets, this book is a testament to human ingenuity and the never-ending pursuit of understanding the very nature of life itself.